hi and welcome everyone to today's coastal state discussion. My name is Meredith Haas and I'm part of the Sea Grant team and I will be your host for today. And before we get started, before we get started on today's discussion, I just want to quickly go over how the flow of today's webinar is going to go. So I will provide a brief introduction of our guest speaker, who will provide a 60-minute presentation, which will be followed by a Q&A with a guest panelist. And so if you would like to submit a question, just please find the Q&A icon at the bottom of your screen, where you can click on it, type in your question, send it out, and we will answer as many questions during the Q&A period as we can. And we are recording today's webinar, which will be posted on Rhode Island Sea Grant's website, as well as we'll be sending out an email notification for everyone who's registered today. And for those of you who are new to the Coastal State Discussion Series, it's an annual event, which is sponsored by Rhode Island Sea Grant, and it's a dedicated platform to sharing ongoing research focused on marine issues impacting Rhode Island's coastal communities. And you can find more information on Rhode Island Sea Grant's website about the series, as well as updates on upcoming events, as well as content from past discussions and lectures. And so on behalf of Sea Grant, I just want to thank our supporters from the University of Rhode Island's Coastal Institute, as well as the College of Environment and Life Sciences and the Graduate School of Oceanography. And so now, having said that, I would like to introduce today's guest speaker, Matt Bertin, who holds a Ph.D. in biomedical science from the Medical University in South Carolina. And he is currently the assistant professor in the University of Rhode Island's Department of Biomedical and Pharmaceutical Sciences, where he specializes in the characterization of marine natural products, algal toxin monitoring, and secondary metabolite biosynthesis. So for today, he's going to be sharing with us some of the work he and his lab have been focused on regarding algal blooms and the associated production of the neurotoxin domoic acid in Narragansett Bay, as well as research investigating how some toxins from algal blooms may be applied into developing drug leads to treat diseases such as cancer. So welcome, Matt, and thank you for joining us today and sharing with us some of this really interesting research.
Meredith, thank you so much. I certainly hope everyone can hear me. I know my video is disabled. Maybe that's just because I haven't shaved for a couple months here, but I'm really, really excited to be here. I wish it could be in person, but I'll just begin. So I really have two stories to tell today. The first story is going to be about harmful algal bloom and toxin monitoring that we're doing in Narragansett Bay as part of a Rhode Island Sea Grant funded project. And then I have a second story where we're going to use some of the toxin monitoring tools that I'll introduce in the first story to investigate HABs, or harmful algal blooms, for new potential drugs. And I think these topics fit really well together. But first, I want to go over for everyone what are HABs. So HAB is an acronym that stands for harmful algal bloom. And I like to break this down by going backwards. So I'm going to start with bloom, and then move to algal, and then move to harmful. Okay, so a bloom. Well, it's a rapid increase in the number of algal cells in a population. And typically, these algae are living in the water, but we can't see them normally because they're microscopic. And when they replicate and grow much larger as a population, then you can actually see these. You can see these even from aerial images. So we're actually looking at Lake Erie. And this green coloration is a blue-green algal bloom that's taking up quite a bit of the lake. And actually, so this is around Toledo, Ohio, and I'm actually from Cleveland, Ohio, right over here. So the algal part, well, algae are sometimes referred to as aquatic plants. They live in the water. Typically, they're multicellular or unicellular, and they generally don't have different parts. So we might have macroalgal species like ulva. Actually, that's a species that some people enjoy eating. But there's also microalgae. So these are single-celled algal species who are the predominant cause of harmful algal blooms. So the microalgae are generally the causative organisms of these HABs. There are many different types. They fall into both eukaryotes and prokaryotes. And I would say that the main groups that are causing the most trouble are the dinoflagellates, the diatoms, and the cyanobacteria. So the dinoflagellates and the diatoms fall into the eukaryote group, whereas the cyanobacteria fall into the prokaryote group. And pictured here at right is a really high resolution electron microscope image of a dinoflagellate. Its name is Corynea brevis. It's responsible for what you may have heard of as the Florida red tides. It produces a toxin called brevetoxin. And the flagellate part comes from this flagellum here, this tail. And then the harmful part. So how do these blooms cause harm? They do in several ways. One is that they can deplete oxygen actually in the water because they're so high in cell number. A second is actually physical contact with other aquatic or marine organisms. So they can attach to, or they can basically clog a fish's gill and cause it trouble that way. But probably one of the biggest harmful aspects of these algal blooms is the production of toxins. And these toxins are organic molecules produced by some algae, which can cause harmful effects. How do they cause harmful effects for us? Well, it can be through the consumption of contaminated fish and shellfish that have the toxin inside of them. It can be from drinking contaminated water with toxin in it, or it can be from recreating in contaminated water. So there are toxins that can give you a dermatitis or a skin rash. And the algal toxins come in an amazing amount of variety. We can see here, I'm hoping you can see my mouse, anatoxin A. This is a very small molecule produced by some cyanobacterial species. You also have the microcystins. These are a bit larger. These are termed cyclic peptides. These are liver toxins associated with, again, those blue-green cyanobacterial blooms in Lake Erie. And some of these toxins can be extremely large. This is palytoxin. This comes from a dinoflagellate. And this is one of the larger small molecules. It's kind of like, I guess, jumbo shrimp. So it's still a small molecule, so it's under 2,000 molecular weight. This might be actually a little higher than 2,000 molecular weight, but around 2,000 molecular weight. So basically small molecules are not proteins, but it's very large for a small molecule. And HABs, they've been referred to as red tides. You may have heard that term. Here are some old newspaper clippings. This is from 1997. This is in Australia. This is a certain species of algae here, and it has pigments inside of it that have this red color. And so that's why you're seeing that very strong red color. And as wave action and water currents move it to the coast, it kind of comes in with the tide, and there you go, red tide. And they can be very large, and they can cover a very large amount of the ocean. You can see a second red tide here. And again, here's the causative organism, these single-celled algae. But they're not all red. They can even be bright green. So again, these are the cyanobacterial blooms that have just been plaguing Lake Erie. And you can see just how thick and just how bright green they are. And they can be large. So I showed an aerial image earlier of Lake Erie, and here's a second. And here's right around Toledo, Ohio, in the Toledo, Ohio watershed. They've had many, many problems with these blooms, even affecting the drinking water of the residents of Toledo, leading to a drinking water ban a few years ago. So they can be large, and they can be very large. So this is actually another cyanobacterial bloom caused by an organism called Trichodesmium. This is the northeast coast of Australia at the bottom left. And the algal bloom can be seen as this sandy color all over the Coral Sea. And these Trichodesmium blooms can cover many, many kilometers. One of the largest HABs ever recorded was caused by a diatom. Actually, the diatom that we're going to be talking about in the first part of the talk. This diatom bloom occurred on the west coast of the United States. So here's Alaska, moving down the coast here into California. And this organism produces a neurotoxin, which we'll discuss in great detail as we progress through the talk. They called this the blob, and we will be discussing it at great length. And there are many different types of harmful algal blooms that are problems in different parts of the country. And so, like I said, we have cyanobacteria. They're primarily found in the Great Lakes region. Also, you find them in Florida. And then we have different descriptions of the toxin poisonings here in this chart. So you might have amnesic shellfish poisoning. That is the result of taking in the toxin domoic acid, which we're going to talk about. And you can see that the amnesic shellfish poisoning and the paralytic shellfish poisoning, which comes from a dinoflagellate and certain species of cyanobacteria, are very common here on the West Coast. And if we go over to where Rhode Island is, we have that problem a little bit as well. So the first part of the talk here, I'd like to focus on monitoring toxins. So we're going to be talking about the neurotoxin domoic acid and the diatom pseudonychia. And because I have such a tough time saying nichia, I'm going to call it PN for most of the talk. When I say PN, that is this diatom. And this is the molecule domoic acid. So when I go out in Narragansett Bay, I'm usually down at Rome Point looking for seals or maybe fishing. So I'm thinking about larger organisms. I've always wanted to go whale watching off Massachusetts. But now we really want to focus on what we can't see in the water. We want to focus on microscopic organisms like algal cells here and here, perhaps even bacterial cells. And we know that there are viruses in the water as well. So we're really immersed here in this microscopic world. And we're focused on these diatoms. These are single-celled algae. They are eukaryotic. They are prolifically photosynthetic. So they are responsible for producing, or they're responsible for about 20% of the primary production on Earth. And they have these beautifully rigid silica-based cell walls. And so some different pictures of these silica cell walls are pictured here. And when I was first starting research, I worked on sponges, and I used to dissolve the sponge tissue in bleach. And the sponge also has a silica skeleton. And you can identify a sponge based on this silica skeleton. And I would always get really excited because often when you would look at the sponge skeleton, you would also see diatoms that were living in the sponge, and they were always a real treat to see. So the diatom we're going to be talking about, PN, pseudonychia, is a chain-forming diatom species. So this is a single cell that I'm circling here, and it can be attached to a second, a third, and a fourth, forming these chains. They do have predators in the bay, which include copepods, which are pictured here, which are small invertebrates. They are capable of producing the neurotoxin domoic acid. And again, this is a high-resolution scanning electron microscope image provided by Dr. Lucy Miranda, which just shows just how beautiful and complex these skeletons are. There are 52 known species of PN, and about half of those have shown the capability to produce the domoic acid toxin. So domoic acid. So how are we as humans getting domoic acid? Well, shellfish that we enjoy consuming, like mussels, clams, and sometimes oysters, can accumulate DA by direct filtration of plankton in the water. So the PN is in the water column, and as these shellfish are filter feeding, they are trapping cells in their gut. And those cells could contain the domoic acid toxin. And if we consume those shellfish, because we're eating their whole bodies, we're typically not, filleting them like a fish, we could be taking in what's in its gut as well. And so the domoic acid causes this poisoning, what's been referred to as amnesic shellfish poisoning, or ASP. The symptoms include nausea, vomiting, abdominal cramps, diarrhea, severe headache, and loss of memory, giving it the term amnesic. And there's a very interesting mechanism of action, which I will explain on a later slide. But the DA binds to receptors in your brain. It ends up releasing massive amounts of calcium into your brain. It basically turns your neurons into overdrive, and it causes vast neuronal damage. And what's very interesting, which is still a mystery, and one of the reasons we love science, is the ecological role. Even after all the years of knowing about DA, it's still not fully understood. And again, the domoic acid structure is it right. It's a fairly simple molecule. You can see the nitrogen-containing ring here, and then you have 1, 2, 3 carboxylic acids. It's very similar to another algal metabolite called canic acid. And they actually act on your nervous system in fairly similar ways. You can see, if you look at the eastern ends of these molecules, they are quite identical. And these poisonings, as a global phenomenon, they really occur at pretty strict northern and southern latitude lines. So if we look at where these red dots are, we can see that they are pretty well restricted here in the northern hemisphere. And there are fewer poisonings, but you can see they're somewhat restricted in the southern hemisphere as well. And since the initial deaths, which I'm going to talk about in the next few slides, from amnesic shellfish poisoning in 1987, because of the intense and widespread monitoring efforts to detect the toxin and the algae, there have been no further deaths that we know of from domoic acid. Okay, so what was the original intoxication event like from domoic acid? So it was late in 1987. There was an outbreak of shellfish poisoning that occurred in eastern Canada. Around 200 people were affected. And they had the exact symptoms that I mentioned earlier. And there were 4 deaths from those 200 people that were affected. And so it was termed amnesic shellfish poisoning, due mostly to the memory loss. And this was a unique constellation of symptoms. Folks hadn't seen anything like this with respect to these toxic poisonings. And so, we're learning, unfortunately, we're learning a lot about epidemiology currently with the COVID pandemic. Well, epidemiologists were called in as well for this seafood intoxication. So they localized the origin of the toxic mussels to Prince Edward Island in Canada, which still is and was an area that's very well known for its shellfish industry, especially mussels. And using a combination of biological assays and spectroscopy. So spectroscopy are a series of experiments to define what an organic molecule looks like. A Nova Scotia group led by Jeffrey Wright, they identified this active neurotoxin in just 4 days. That is an unheard of feat nowadays. I'm going to show a series of molecules we've isolated from my laboratory, and I can assure you that it took more than 4 days to figure out what all of them looked like, probably more than 4 weeks. And so they were able to do this. They did it in a different way than we normally do it now. So they found this domoic acid in the mussel tissue that they were able to collect from Prince Edward Island. And they had some advantages while they did this investigation. There was a very large amount of the toxin in the affected mussels. And so they had a nice group of non-toxic control mussels to compare what they were finding. Instead of using cell-based assays like we do now, they actually were separating out the mussel extracts into different chemical groups. We'll talk about this type of method later in the talk. And as they separated out these groups of chemicals, they were actually injecting them right into mice. And they were looking to see if the mice had those same type of neurotoxic effects. And that's what they used to guide them for the ultimate isolation of the molecule. It was also aided by the water solubility of the molecule. And so domoic acid was not an entirely new molecule when they found it in 1987. It actually had been described in 1958 as a product from a red algae in Japan, but its toxic effects were not known at the time. Okay, so how does domoic acid work in the human body? Well, if you look at the left side of the screen here, the domoic acid, the nitrogen, and these two carboxylic acid groups, they look very, very much like glutamic acid. In fact, if you put your hand over this left side, they are identical. And so glutamate, or glutamic acid, it is a very important neurotransmitter compound. And so the glutamic acid is packaged into vesicles in your neurons. And you have a presynaptic neuron and a postsynaptic neuron. And the pre is sending a nerve signal to the post so it can continue, and you can carry signals throughout your central nervous system. So as the glutamic acid molecules are released, they are hitting receptors. So they're released from the presynaptic, they're hitting receptors on the postsynaptic. When they hit those receptors, calcium is released, and calcium can continue on as an ion to propagate electrical signals. Okay, so what does domoic acid do? Well, when it gets into your body, these receptors can't differentiate it from glutamic acid. They think it's the same thing. So now you're getting this very large dose of glutamate. You're having an uncontrolled, basically, release of neurotransmitter and an unregulated influx of calcium into your neurons. And that's creating a tremendous amount of excitation in those neurons. You're basically overexciting them, and those neurons will eventually die. Okay, so that's some background on domoic acid, its discovery, where it comes from, and how it works. But why are we worried about domoic acid here in Rhode Island? So now I don't have to tell you folks where Rhode Island is, but I like this slide. My grad student found this slide, and I think it's really cool when we do talks in other parts of the state where they're not as familiar with Rhode Island. It's nice to show a map. But we have a shell fishery that is key to our economy and really key to our blue economy. We have $6.5 million tied up in wild harvest. We have many, many commercial shellfishers. We have widespread aquaculture with 70 farms. We have 11 different species of shellfish sold to all 50 states, and it's free to harvest if you're a resident as long as you have a permit. We have these numbers here from 2016 RIDEM. Another thing that we really have that's great in Rhode Island is the long-term Plankton Time series, which is conducted at the Graduate School of Oceanography at URI. So Narragansett Bay is a diverse and productive estuary, and it is so great that we have this access to these long-term data sets, understanding what the phytoplankton community is like, and also understanding what some of the physical water parameters are like in Narragansett Bay. And I believe that this time series has gone on since 1957, collecting weekly samples. It is one of the world's longest running Plankton Time series. So we have quite a bit of information on the phytoplankton community going decades back. And so what's the history of PN in Narragansett Bay? Well, we can see from this chart here, this graph, that Pseudonychia has been present in Narragansett Bay for over 50 years. Well, not all 50 are pictured here, but if you go back even further in historical data. And you can see when we look here on the y-axis at cell counts per liter, and on the x-axis we look over time, we have high numbers of these PN cells. However, the first emergency Rhode Island shellfish closure due to the neurotoxin domoic acid being in the shellfish, that didn't occur till October 2016. And then again in March 2017. So these closures are happening here. And what's interesting, they're happening with cell numbers that are much lower than what has been seen historically. And so that's really where other researchers who are studying this in the Bay and where our research really takes off, it's the presence and persistence of these blooms have led us to, and the presence and persistence of the domoic acid have led us to try to understand what's driving this, what's driving these new toxic events. And so this was covered in the press quite a bit because it is a big deal. We don't want to see these shell fisheries shut down. I myself love eating oysters and mussels and clams. And this made me think of an old paper that I had seen, knowing a little bit about domoic acid. I did my postdoc on the West Coast. And so they have many more events over there. But this is a newspaper from Santa Cruz, California, from 1961. And what we're looking at here is a picture of a police officer. And this is unfortunately a poor seagull who's run into his police car. And what happened was that there was a PN bloom off the coast of Santa Cruz, California. And the shellfish there had been inundated with domoic acid. They were feasted upon by a whole variety of birds, shorebirds, and the birds had the effect of the neurotoxin. So they were disoriented, they had loss of coordination, and many of them were flying into buildings and flying into cars. And believe it or not, this was one of the events. It's not the only event, but it was a large event that partially inspired the Hitchcock film, The Birds. So I'm not sure if anyone knew that, but there's some trivia for you. Okay, so how do we come into this research? Well, what we were really interested in was to kind of get at the first principles of the PN community in the Bay and the production of domoic acid in the Bay. We had some initial questions that we were interested in. Are there new toxic PN strains that have somehow come into Narragansett Bay that weren't here before? Or are there resident strains that have become toxic because there has been a change in certain environmental parameters or perhaps anthropogenic changes, man-made changes? So how are we going to do this? How are we going to carry out this research? Well, we wanted to identify the Pseudonychia strains in the Bay using molecular means. We were going to record temporal and spatial variations of these strains and on domoic acid production. And we were going to try to collect as many environmental metadata points as we could. So we wanted to know every time we went out to sample, what was the temperature of the water? What's the pH of the water? What's the salinity of the water? What is the phosphorus concentration of the water and the nitrogen? We wanted to be able to learn as much about the environmental parameters that were occurring in the Bay so that we could hopefully find some links between these parameters and the possible production of domoic acid, or perhaps the possible effects of these environmental parameters on how certain strains can become more abundant. And we had 3 major ship-based sites or vessel-based sites that we would visit. We had, again, that GSO time series that's been just so valuable. And then this is in the Upper Bay. And then we had 2 other sites, one at Whale Rock, which is in the West Passage, and then one that's in the East Passage, which we call East Passage. And we tried to get as much data from the vessel-based sites as possible. We also had some shore-based sites. We had one near Castle Hill Inn, one at Fort Wetherall, and one at the GSO dock. So when we go out, we take an instrument called a YSI, and this instrument is going to be able to measure many of these environmental factors for us: the temperature, the salinity, the pH, the depth, the chlorophyll content, and the dissolved oxygen. And we're going to collect basically a bucket of water. So we're going to collect a water sample from every site that we go to, and we are going to look at the genetic information of the diatom community and the PN community. We're going to look at the amount of domoic acid that is in the phytoplankton cells. We are going to measure how many cells there are at our sites, and we're also going to take a nutrient sample. So we are doing this every week. And we've been doing this every week for over 2 years. So the first thing I'd like to do is talk about the identification of the PN species using genetics. So full disclosure, I'm really more of a chemistry person. I've done some molecular biology back in my career, but that's why I have such great collaborators who can really help me out. But I wanted to introduce the basic workflow of how we're trying to do this. This is a sample from, or an example from bacteria, but the same basic process is being done with the diatoms. We're just using different genetic markers. So we use a DNA barcoding technique, and how we utilize this technique is that we can collect an environmental sample. So here we're showing bacteria, but we're collecting phytoplankton, and I'm sure that, well, we know there are bacteria in it too, because it's out from the environment. So we're collecting this sample, and we're going to do that by filtering the water that we collected, and I'll show that in one more slide. And we're going to extract the DNA from this filter. So we're going to have a community of DNA. We're going to have diatoms, we're going to have bacteria, and other organisms. Then we're going to apply PCR primers that are going to express stretches of specific genes. And we are going to sequence those genes. And we are using diatom-specific primers here, not bacteria-specific primers. And we can amplify small stretches of conserved genes in these organisms. And we can compare the genes that we're finding from the diatoms in the environment to that which is known from different databases. We can do phylogenetic comparisons, so we can see the strains that we're finding in the bay, how closely related are they to other diatoms that have been found, other species of PN that have been found. And we can get a really good sense for community composition by, again, using the genetic markers to discriminate between species, but also using the amount of sequence that we find to infer the abundance of different species. And so how are we doing this? We are taking that bucket of water, we are filtering it on a 5-micron filter. That filter is extracted for DNA, and then we are using conserved genes that all species of PN have for PCR analysis, and ultimately PN identification. And so what does it look like actually in the lab? It's pretty interesting, intricate process. So we are taking that whole water sample. It is being filtered using a peristaltic pump. There is one inline filter here, which is 5 microns, which is collecting the phytoplankton, including the diatoms. That water is passing through. It's hitting a 0.2 micron filter, so that's much smaller than 5 microns. And bacteria are much smaller than diatoms. So this filter will capture the bacteria. And then the water will flow through, and this can be used for nutrient analysis. So from this one filter, we get many samples from which to analyze. So we can analyze the PN species molecularly. We can identify and analyze the bacterial species genetically. And then we can measure the different nutrients that are in the water. And instead of using that 16S gene that the bacteria have, we're going to use the 18S gene that eukaryotic organisms have. We're going to use this V4 region, a small region on the 18S gene, and this will help identify PN species. Now, we found from our work that we think that using a different gene sequence here, this ITS1, that this will give us even better resolution, that it is a better genetic marker to determine the PN community composition. But we have data for both, and we can actually compare them. Okay, so we did our first barcoding with the 18S gene. We were able to show that the PN community composition did change seasonally, but we wanted to see if our species resolution could be improved by using this second genetic marker, which was the ITS1 region. And we think that this is going to give us some superior ability to discriminate between these PN species or among these PN species. And what we really want to show, and I have one of these later in the talk, just one of these hot off the bench slides, which I haven't had a lot of time to interpret yet, but I think it's so cool that I think it's worth sharing. And what we want to do here is to determine the variability in our Synonychia communities, and then integrate this with the domoic acid data that we're generating. And we're going to try to develop these really data-rich, long-term time series data sets that I think are really, really exciting. And they're coming up in a few slides, and I really can't wait to show them. Okay, so what do some of these data pieces look like? So if we, and sometimes I switch back from ITS sequences to 18S, so I'm going to try to make sure that I'm saying when I'm doing that. Okay, so we are able to determine what the diatom community composition, the PN community composition is over time here. So we can look at a time point in October of 2017, and we can see that the sequences that are dominating in our analysis are P multiseries. If we look at a further time point in January 2018, we see Americana and we see Delicatissima. If we look at April 2018, Delicatissima and Pungens. In July 2018, Delicatissima, multiseries, and Pungens. And so we obviously still are waiting on some data here. We want to fill in our entire 2-year project period with all this genetic data. But the take-home message from the slide is that this PN community does appear to be changing in composition over time, and there is often more than one strain present. Okay, so we feel that we're totally on the right track here. We're finding really interesting information on the PN community. But another big part of what we wanted to do was understand the production and the concentration of this neurotoxin out in Narragansett Bay. So how are we going to do this? Well, we're back to taking that water sample and filtering it for analysis. So much like the DNA filter, we have a second filter. And we collect the phytoplankton cells, including the diatom cells, on another 5-micron filter. We extract that in 0.1 molar acetic acid. And then we use something called a mass spectrometer, which I'm going to explain in a moment, to analyze the toxin concentration. And we use a very interesting technique called LC-MS-MS with MRM monitoring. And I'll try to explain that as best I can on the next slide. So, okay, how do these instruments work here? So why is a mass spectrometer so powerful? Well, we are extracting this filter, right? The filter has phytoplankton cells all over it, many different phytoplankton cells. So when you extract it, you're going to have a mixture of compounds. This is a stock photo that I had. It's not purple, it's green. But you have this mixture of compounds. You have many, many chemicals in here from the phytoplankton. And we can send this to something called a liquid chromatography system. So that's LC. And what this system is going to do is it's going to separate out individual compounds that are in the mixture. After we separate them, we send them to something called a mass spectrometer. The mass spectrometer, basically at the end of the day, is allowing us to infer the molecular weight of all the compounds in our sample. And how does it work? It's a very interesting system. The liquid is sent from the LC, and it's sent into the mass spec, and it's very, very hot, and there's a stream of nitrogen. And basically, you're taking the liquid and you're vaporizing it into gas, and you are charging it into ions. These ions flow through a quadrupole, so they're sent down to a detector, and this detector is basically giving you individual numerical measurements. It's basically telling you how much these molecules weigh. So to go into it just a little bit more, you have, again, the LC system. You have this column in the LC system. You inject your sample, it goes over the column, and these individual molecules are separated. Now, one thing we do to ensure the accuracy of our measurements is we run a domoic acid standard. And we know that our domoic acid standard comes out at 11 minutes. In addition to running the standard, we also employ this MRM monitoring. So what does this really mean? Well, the way I would explain it the best that I can is that if you have a crude sample, right? So we extract that phytoplankton, and there are many, many molecules in the phytoplankton. And we're really just looking for one. We're really just looking for this domoic acid. Okay, well, if we looked at all the molecules, we'd see many different ions popping up, many different weights, let's say. We'd see 300 and 400 and 500 and 600. We don't want to look at all of those. We only want to look at molecules that have a mass to charge that show up in the mass spectrometer with 312, which corresponds to domoic acid. But even 312, maybe there's more than just domoic acid that has this number of 312. Okay, so we do something extra, which this special mass spectrometer allows us to do. We fragment molecules that have a weight of 312. And we only look at fragments that theoretically would come from domoic acid. They're called transitions. So we look at molecules at 312 that when we fragment them, we give them extra energy and they fall apart. So when we give this molecule extra energy and it falls apart, if it falls apart and we see 266 as a fragment, 247 as a fragment, and 193 as a fragment, well, there's only one molecule that's going to really fragment like that, and that's going to be domoic acid. So that was kind of a long spiel there to try to explain that. But the take-home message is that this type of analysis, this LC-MS-MS with MRM, it gives us very, very good sensitivity. We can measure low levels of domoic acid, and it gives us absolute accuracy. So we are quite sure that we are measuring domoic acid. And then also we have our nutrient analysis. So again, we have the filter, the inline filters. We collect the diatom for the phytoplankton analysis. We capture the bacteria with a smaller pore size filter. And then the remaining water that elutes out, we analyze that for nutrients. And why are we looking at nutrients? Well, we surmise, we hypothesize that toxin production could be due to a limitation in key nutrients that are required for growth. And so phytoplankton need nutrients like most species of phytoplankton for sure need nitrogen, phosphate, silicate, calcium. They consume carbon dioxide, they release oxygen. So we know that these nutrients are going to be important. Okay, so now I think we get to some really fun stuff here. And so I'm going to try to take this as slow as I can. I can tell I'm getting a little excited here because I really like this figure. But we have 3 panels. I'm going to go to the bottom panel first. We have the PN cells that we've counted over the entire length of our project period, going from 1017 to 2020. The dots that you see correspond to our different sites here. So Whale Rock is light blue, East Passage is yellow, and the time series site is dark blue. So we have our PN cells. We have our domoic acid, our cell-associated domoic acid from those phytoplankton filters. We have nanograms per liter of seawater there. And then we're also looking at combined nitrite and nitrate measurements in this figure. And so what are some of our big take-home messages that we've been able to find over this 2-year project period? Well, I think one of the most interesting ones is some seasonality to when we see spikes in PN cells and spikes in domoic acid. So here we're looking at June 2018 and June 2019. And we see cells come up and we see cells come up. And we see corresponding increases in domoic acid coming up and domoic acid coming up. We also see, whoops, going the wrong way. We also see fall seasonality. So we have September, October of 17, September, October of 18, and September, October of 19. We see not the same peak in cells, but we do see these high spikes in domoic acid. We don't quite see the same in 2019, but there still is an increase for sure. And this has not only been observed by our group. There does seem to be a decoupling where you can have low amounts of cells and high amounts of domoic acid toxin. We're not exactly sure why that is. It could be that these fewer number of cells are producing much more domoic acid because of some type of environmental reason, but this is something that we're very interested in finding out. And so again, if we look now, we have that seasonal pattern of fall. Then, so basically September, October, June, September, October, June, September, October. And one thing we're noticing, and again, we are not trying to infer too much from the data at this point. We're just trying to identify patterns that we can explore further. But we do see that during periods of low nitrite and nitrate is when we do happen to see the most toxin. So here we have increased levels, you're going down, you see 2 periods of toxin coming up, you have nitrogen coming back up, going back down, and now you're seeing toxin again. So this is something that we definitely want to explore further, but we can't make too many claims on it right now. And again, one area where I really see this being very powerful. And again, this is this hot off the bench data that I was talking about. But what we eventually want to do is, you know, I just love this figure here with these different panels. What we also eventually want to do is be able to look at the genetic information that I showed earlier and be able to examine the PN community composition to see if it changes over time and to see if that somehow reflects with toxin composition that we're seeing. So we have these different sequences here. So basically, if you see a different color, it's likely that that could be a different species or strain of domoic acid. And so really the only thing I can say about this right now is that we definitely see species composition change over time. So, you know, we have the red color here, a lot of blue for a while, the green and red color. And so we really want to dig into this, and we really want to try to integrate this in with the toxin data. This is just for the Fox Island site right now, but we should be able to do this for the East Passage site and the Whale Rock site as well. So I do think that this is really kind of one of the big keys here for where we want this data to take us. Okay, so one last story here. During the June 2019 DA event, we did go out and collect some mussels. So here, this is capturing basically the DA dynamics. So if we're looking here at Whale Rock, you can see that's coming up around June 1st. I think the peak was around June 4th, and then it's coming back down here, and it's basically following that at all our sites. So on, I think it was June 4th, we were able to go out, we collected a few mussels, we extracted those mussels in a similar way that we extract the phytoplankton. And we did that same mass spec analysis. We were interested to see during a DA event, would we find any in the mussels? So we did examine 6 mussels, a very small amount of mussels to examine. And we're only looking at each of the single mussels and the concentration of DA. So we did find some domoic acid in the mussels. However, I think it's important to stress these levels were far below the National Shellfish Sanitation Program's threshold, which is really between 10 and 20 micrograms per gram of shellfish tissue. That's what could trigger a mandatory closure. We're talking about 4 nanograms per gram. So we're talking like 1,000 times below where these concentrations cause shutdowns. So we were just interested to see, would we find some DA in the mussels, but they're nowhere near closure levels. Okay, to summarize where we're at here, we find that domoic acid production from PN, it's been emerging problems in 2016 in NMB and Narragansett Bay. We are showing that the community composition is differing seasonally, and also that DA production shows seasonal trends. The pseudonitria cell counts and the domoic acid production do not always correlate. That's something that's been a mystery for quite some time that we're very interested in. And we have some preliminary nutrient analysis that shows that some domoic acid events are occurring after periods of low nitrogen. But I really want to stress that we're just at the beginning of compiling and integrating our data together. So some future plans, again, to really dig in into some of these nutrient regimens and see what we can infer from the data we're collecting. Probably I say our biggest goal right now is to get unialgal PN isolates in culture. we want them to be toxin producers, and we want to be able to take the environmental parameters we are finding from our collections, and can we perform experiments in the lab, controlled experiments in the lab, to see if we can correlate increased DA production with possibly the limitation of certain nutrients. We've also been looking at some archive samples from earlier, trying to look at what that PN community looked like. And we really want to continue to collect our time series data. We think we're finding some, some pretty interesting things. So I want to do an acknowledgement slide for the first part of the talk right away. Really like to thank Rhode Island Sea Grant, CAIM, RI Space Grant for continued funding. I'd really like to thank the co-investigator, Dr. Bethany Jenkins. Her lab does just so much. They're just so helpful with everything. Alexa Sterling and Riley Kirk are grad students who have really just kind of driven this project. We have a whole collection of other grad students and undergrads like Catherine and Megan and others who have just done such a great job. Dave Borkman at DM's and really great to work with and talk with. Lucy Miranda, Jessica and Nina collect a lot of water for us, done a lot of work, and Steve at Barber and Granger for captaining. So now I'm going to switch gears, and I'm going to try to make this move into how my laboratory is really interested in trying to discover new drug leads from these harmful algal blooms. Now we're not talking in the diatom space anymore. We're moving over to cyanobacteria. And so we're going to talk about some small molecules. There's going to be some chemistry, but not too much. It won't be organic chemistry class. So cyanobacteria are very important in the environment. They're a phylum of bacteria, even though they are often referred to as blue-green algae, but they are true bacteria. They're photosynthetic autotrophs, so they photosynthesize, they make their own food, they get energy from light, and they produce oxygen. And what's really interesting is they are thought to have been responsible for an oxygenation event that created the oxygen-rich environment that we all enjoy nowadays. So we can thank cyanobacteria for that. That was about 2.5 billion years ago. And they produce toxins and other organic molecules. They occupy diverse environments. You'll find cyanobacteria in fresh water, like the Great Lakes, like Lake Erie. You'll find them all over the ocean, and you'll even find them on land. Have you been down to the Newport Cliff Walk? Well, if you have, you've probably seen this growth on the rocks. This is a lichen, and a lichen is a really interesting symbiotic relationship between a cyanobacteria and at least one fungus, and it might be even two fungus, or I guess it'd be fungi. So we're walking by cyanobacteria all the time, so we're all enjoying them. You can even find them in the Arctic. But do cyanobacteria make compounds that can become drugs? And they have. It's hard to get a drug FDA approved, so there are only so many great winning stories in that realm. But I think one of the most interesting stories in the history of drug discovery, not just from cyanobacteria, but really from anywhere, is the story of a molecule called dolastatin. So dolastatin was originally discovered decades ago. So natural products chemists, so folks who like to go out into the environment and make collections and see what type of chemistry is in an organism, and to see if that chemistry can ultimately be leveraged and turned into some type of pharmaceutical, were going out all the time and basically collecting everything they could find in the ocean. And one thing they were looking at was an organism called a sea hare, or a nudibranch, pictured here. And so they went out, they collected a lot of these nudibranchs, and they found this organic molecule. It's called a peptide. We don't have to go into it too much, but this molecule was extremely cytotoxic. It was very, very toxic. And they tried to make it into a drug for a very long time. Then years later, someone found that the same dolastatin molecule was found in one of these cyanobacteria that you can see here in this filament. So the same exact molecule was found from the cyanobacteria and found from the sea hare. Well, it turned out that the sea hare was eating the cyanobacteria, and it was concentrating the dolastatin in its body to ward off predators. So how cool is that story? And it took many, many years later, but I think it was 2015 or maybe 2014. But this molecule was ultimately turned into an antibody drug conjugate. That's a specific type of drug, and it's used against cancer. And it's called brentuximab vedotin. So, a really great story when it comes to cyanobacteria. Okay, so how are we fitting in with the cyanobacteria? Well, we are trying to develop these drug leads from, again, these cyanobacterial blooms. And we are really early in the process. So we're in what's called the screening and the optimization phase. We aren't really close to these animal studies or clinical studies. The screening phase is a method or a workflow where we're using different biological assays that are informing us of a possible therapeutic effect that extracts and fractions might have. So what could this assay be? It could be a cytotoxicity screen using cancer cells. It could be using methicillin-resistant Staphylococcus aureus to see if we have any extracts that might stop its growth. It could be an antifungal assay. It really could be anything. And then if we can identify an extract that has this activity, we can break that extract up into all its component molecules. We can isolate the pure molecules from that extract, and we can see how potent those are. Do they do a really good job at killing those cancer cells? Do they do a really good job of killing those bacteria? And that's when we want to optimize these molecules. We might have to change them a little bit. We might have to add some stuff to them or take some stuff away from them so that they can actually move in to these further studies. So I just got to stress we're really early in this process. Okay, so how does this work with cyanobacteria? How does this work with these blooms? Well, historically, what you do is you go out and collect, you screen extracts, or you screen fractions, and then you identify these hits, and you purify out these new potential drugs, and you do further studies. Okay, so I'd like to go out, scuba dive. This is not a blooming organism here. This is just on the bottom, but you go out, you're on scuba, you're reaching down, you're collecting this stuff, you're putting it in a jar, you bring it back to the laboratory, you extract it in some type of solvent. So you're pulling out all the chemicals from that cyanobacteria. Now, often that extract is far too complicated to analyze. There are just too many molecules in it. So you do a process called fractionation. The best way I can sum up fractionation is you are breaking up the crude extract into simpler mixtures of molecules. And at this point, you can, at this point, you can take these fractions and you can test them. You can screen them. So here we screen these fractions in an assay. We wanted to see if they were toxic to brain cancer cells. We want to look at the viability of these cells. Do they grow? Do they grow like the control? Do they grow to 100%? If the bars are lower, like D, at 2 doses, 40 and 4 micrograms per mil, we have very low bars. We have a hit now. We have a fraction that we want to investigate further to isolate possible drug-like compounds. We've had a lot of interest in this other blooming organism, this blooming cyanobacteria trichodesmium. It's a very important nitrogen-fixing cyanobacteria. So it takes nitrogen out of the air as N2, and it converts it into usable nitrogen for other organisms. It forms massive, massive surface accumulations. Believe it or not, James Cook actually documented finding trichodesmium blooms when he was out sailing. They used to think that they were shoals. And to be honest, this was just a very simple question. Nobody's looked at these blooms, so why don't we do it and see what type of molecules they might make? So we went out in the Gulf of Mexico aboard one of NOAA's ships. They were nice enough to take us out. And believe it or not, we went right through a bloom. We got super lucky. So we collected the material. There's me trying to get some of this stuff out of the nets. Okay, so where I want to try to tie in the monitoring and tie in our drug lead investigation is going back to the mass spectrometer. Okay, so we talked about the mass spectrometry, right? We have this mixture of compounds. So now we have a mixture of compounds from our trichodesmium bloom material, just like we had a mixture of compounds from our phytoplankton material in our Narragansett Bay study. We need to separate these out so we can analyze them, and we need to use the mass spec to figure out how much they weigh and maybe what they are. So when we were looking at the domoic acid, we only wanted to find one molecule, but we wanted to do it in a really specific way because we knew that this molecule broke apart in a very specific way. And we could ensure that we were only documenting domoic acid. Okay, so this gets a little technical here and I apologize, but instead of looking for one molecule using this type of technique, what if we looked at all the molecules in a sample? And so what we did here is we used a new tool called MS-based networking. And so what we're doing here is we're looking at our mass spec. And then we're saying that if we fragment all the molecules in our sample, they are going to break apart in a very predictable way. The most labile bonds are going to break off, just like domoic acid fragments in a very predictable way. And if two of the molecules share similar fragmentation patterns, it's like having two similar fingerprints. And so if you have similar fragmentation patterns, we can cluster you together. We can assign you a score. You're 75% similar. You guys, you're only 20% similar, and you're only 10%. And so we can make these networks of all the compounds in these samples. And so that's what we did with trichodesmium. And so here is kind of the, what does that look like? So again, so the take-home message here, these similar compounds fragment in similar ways. And we can cluster those together. They basically have similar fingerprints. And so what does this look like when it's applied? Well, it looks pretty wild. So let me take a minute here. I have about 5 minutes, I think. We have identified hundreds of compounds from the bloom material. What's really interesting about this study is we looked at blooms from 2014, 2017, and 2019. Those are represented by colors in these pie slices. So we're finding many of the same molecules year after year. And this has allowed us to isolate and identify many new molecules. So how does the networking work? Here is a molecule we call trichophycin B. You can see its structure here. And it's very similar to this other molecule here, smetolactone D. And so because these molecules are similar, they cluster together. And we found even more interesting new molecules. So we are very excited by this tool. And we've been able to find over 28 new molecules at this point, which is really great when you're into drug discovery. So I hope there's some researchers out here on this call. If you're ever interested, if you need a small molecule isolated, look us up. We like to do this kind of stuff. But the real question is, do any of these molecules have any promising biological activity? That's the whole point of this, right? So I'm only going to focus on two for just a few more slides. The first is this trichophycin B. And this is a pretty standard approach. Folks are very interested in anti-cancer drug leads. So we looked at trichophycin B, and we looked at two different cell lines, MCF7, which is a breast cancer cell line, and BXPC3, which is a pancreatic cancer cell line. Here, we are dosing with increased higher doses as we go of our molecules, and we're looking at trichophycin B in green. So if trichophycin B is killing the cells, the bars are going to drop, and they do. And this molecule shows some potency, 652 nanomolar IC50. That's a it's not very, but it's a fairly potent compound. It's a compound that actually could continue down this drug pipeline to see if we could really develop an anti-cancer molecule. And it also lowered those bars against the pancreatic cells as well. Okay, so this one, there's a lot more data on this slide, so I'm going to try to take a little bit slower. But the second molecule we looked at was called unarmacin D, and it showed very potent and very intriguing anti-inflammatory activity. So how did we do this? So here's unarmacin D. What we did was we took a microglial cell line. So microglia are like the macrophages, like the immune cells in your brain. And we're very concerned with neuroinflammation, right? If you have traumatic brain injury, you get neuroinflammation. If you have neurodegenerative diseases, you have neuroinflammation. So lowering neuroinflammation is very desired therapeutically. It's associated with reducing pain. So what we do is we take the cells and we subject them to bacterial LPS, and that creates an inflammatory response. And then we have dosed our cells with unarmacin D, and we want to see these bars lower. We want to see the nitric oxide content, which is a pro-inflammatory molecule, go down. And with unarmacin D, we did see it go down. We also saw other cytokines. So these are other small molecules that are released during the inflammatory process. And so unarmacin D, this is its code here, the GH2C1. Unarmacin D decreased the inflammatory cytokines as well. So we were really excited by that. So it was showing good anti-inflammatory activity. And then we got really, really excited because we found that this molecule bound to both the mu and the delta opioid receptor. And so these receptors are very important drug targets because we have a big problem with the opioid epidemic. And so we want to find molecules that are not addictive in the same way that our current opiates are, but that also can provide some pain relief. So again, this is really exciting information. I know that was a lot to throw at everybody at the end there, but I'd be happy to talk about it in the Q&A session. So our summary and our future plans with trichodesmium, so we found that these colonies are plentiful in the Gulf of Mexico. So we think we have a really great some field stations out there. And I hope I was able to convince you that because we looked at the 2014, 2017, and 2019 samples over time and at different geographic locations, we're finding these compounds year after year. So we think that's really cool too. And we can look at them all when we make these networks. And so what are our continuing efforts trying to take these hits to leads? We want to know how selective and potent trichophysin B is. So is it only effective against MCF7, breast cancer, and the pancreatic cell line? Is it toxic against all cancer cell lines, or maybe just a few? That's something we really want to know. What we want to know with unarmacin D, are they agonists or antagonists? How do they actually act on the receptors? That's really important to know if you want to try to make drugs. And can we change unarmacin D a little bit? Unarmacin D is a peptide, so it's actually kind of easy to change it. And so we might be interested in trying to make something that binds a little bit better or shows a little bit better anti-inflammatory activity. So we really think that there's a lot here in the realm of drug discovery with these blooms, and we think using some of these fun tools that we used in monitoring can really be applied well in drug discovery. So to acknowledge this part of the project, I'd really like to thank grad student Chris Vi and also Riley Kirk again. We've had, again, very helpful undergraduates. The folks at NOAA were so great. They picked us up from the airport. I thought that was the coolest thing ever, so we didn't have to take a cab. Alfonso Mangione, he's at the University of Naples. He's a collaborator of mine. He helps us work on those networks that I showed. And kind of just a quick interesting aside on Alfonso, we had never met before. We only talked over email. We had written a paper together, and we didn't meet until February when we went to a Gordon conference together. So I thought that was really interesting. And some folks at Texas A&M have been really helpful in collecting samples for us over the years. And so with that, I really just like to say thank you. I hope this is going well, but I'm going to stop talking, and I'm very, very happy to take any questions.
Thanks a lot, Matt. You should have video capabilities now. Hopefully we have that working. But yeah, again, for folks that have questions, please just go to the Q&A icon below and type it in. We do have a few that I'll relay right now. The first few really are related to the first portion of the presentation concerning domoic acid. So first comment question is, it has been proposed that phosphates have a stronger effect on harmful blooms. Why did you focus on comparing seasonal patterns of domoic acid versus nitrate? It would be more interesting domoic acid versus phosphate concentration.
Yeah, that's a very insightful question. It has probably the first answer is the simplest answer. We just got to our nitrate levels first. It was just some things we were first starting to analyze. The phosphate recommendation, again, it is very insightful. There's been some really interesting work that came out, I think it was last year in Science, on the biosynthetic gene cluster, so the genes that actually form domoic acid in the organism. And one of the drivers that led them to identifying these genes was culturing the organism under phosphate limitation. So they decreased the amount of phosphate that the organism was able to use, and the genes for the toxin basically turned on. So yes, I do think that it would be very interesting to focus on phosphate. We do have these data, and we are going to be spending lots of time here analyzing that. So it's a well taken it's a well taken question. So,
I want to point out something. I had a question and then you kind of answered it within the presentation, because, you know, something we had talked about earlier too, in terms of nutrient levels, potentially being triggers for domoic acid production. And one of the questions I was thinking of, I'm like, oh, well, do you need a bloom necessarily to have a toxic event? And it seems like that's something that's still under question, that you don't necessarily need to have the cell counts to have that toxicity present.
Yeah, it's a mystery to us still. You know, we're not exactly sure if it has something to do with persistence of cells in certain areas of depth, you know, that we could be possibly missing. You know, we're all ears for what other folks are finding as well.
All right. Well, the next question is, in earlier pseudonychia events, which extended from Maine to Rhode Island, I think that's referring to the 2016 event, pseudonychia, or not pseudonychia, but P. australis, am I pronouncing that right?
Yeah, Australis. Yep.
Australis was identified in some locations. This was the first time since people started looking at that this species was observed. So it's hypothesized to be the cause of the sudden increase in the domoic acid production. But your data suggested that Australis multi-series. So why do you assume that it's the multi-series species? Because it is also toxic, but just not as much as the Australis.
Yeah, I think we're waiting fully for our sequencing information with the ITS results to try to resolve some of that with the Australis multi-series. I know, I think I'm allowed to say this. You know, I've communicated with some folks on the West Coast, and I think Australis could be something they're finding as well. So I think we're going to wait for getting the full kind of global complement of all of our sequences to really parse through those. But again, that's a very insightful question too.
Okay, and so let's see. Then the next comment is, domoic acid contains a considerable amount of nitrogen. The factors affecting domoic acid concentrations, either intra or extracellular, are complex, but tend to be high when nitrogen is available, especially if phosphate or silicone is limiting. So I wonder if phosphate or silicone is more limiting than nitrogen, even if all are low.
Yeah, I mean, that's, again, yeah, these are extremely insightful questions. Yeah, I think looking at ratios is going to be really important as well. And so I can't stress enough how early we are in that process. But again, you know, this careful introspection of data is something that we're really geared up to do. But I mean, yeah, I think every single question here is really fantastic.
Well, let's see. The next one is, I always had the idea that harmful algal blooms are propelled after hot seasons, which makes sense with your seasonal plot showing spikes during September, October, but I also see spikes in January. Does that mean heat is not mandatory for blooms?
Yeah, so some of the data we showed might have been a little limited. I'm not sure I agree with the spikes in January. Maybe I don't know my own data strongly enough. I think for all of 2018, we really didn't see much in January in terms of spikes. We may have seen a little bit this 2019, and then 2020 maybe a little too. I guess I need to look back at that slide. As far as being propelled by hot seasons, we don't know enough, I think, from our data set to say that. I don't think we've analyzed temperature enough. But again, that is something we'll be looking at.
Okay, so Bethany, I included you as a panelist because I saw that you made a comment, so I wasn't sure if you would like to speak more to that.
Yeah, Bethany is much better.
You need to unmute me.
Yep, you're good.
Okay, all right, great. Yeah, thank you. I saw some of Kay's questions, and it's great to have you on this webinar, Kay, and everybody else that's tuned in. And I think Matt's done an amazing job of summarizing our project. I just wanted to comment that one of the things that could also be contributing is both a combination of warming water and perhaps stratification. And these are the kinds of variables that we're putting together and sort of trying to get an understanding of. And I think the thing that makes this so hard, and we can start to do this with Matt's domoic acid measurements, is obviously, you know, the presence of pseudonychia doesn't indicate the presence of toxin production. So to have these, I think what's unprecedented, and I'll just toot Matt's horn a little bit, in the kind of measurements that he's making, is these absolutely sort of near real time, very high time series resolved measurements with a very sensitive method that's much more sensitive than other quick methods for domoic acid measurements. And so we're seeing very sensitive methods for domoic acid, which I think will help us really start to untangle the relationship of these environmental drivers and domoic acid production, since we're kind of working in sort of subclosure space for most of this, that having the sensitivity of Dr. Bertin's methods is really going to help us look at these events like warming seawater, nutrient concentrations, and the time series data is going to allow us to sort of look at what was going on in the preceding weeks leading up to some of these events. So I'm really excited to be working with him to put these data together.
Excellent. One question I have, and maybe you answered this, I'm trying to relate this to the discussion on drug development, because we first are talking about diatoms and then we're talking about cyanobacteria. And I'm thinking, you know, are there molecule signatures that lend themselves to drug development? Because I'm assuming that domoic acid, not a candidate in any way, shape, or form.
Yeah, that is an awesome question that I might have to take another hour to answer. I'll keep it brief. So there are, I would say that generally very polar, so there are always caveats to every rule, but generally polar molecules like domoic acid, so very water soluble molecules, have not been desired for drug leads, especially in the anti-cancer realm. I will say really quick, it's not to say that there's no potential for it to ever be repurposed. It would take a lot of medicinal chemistry to make it a good drug lead. But there are other toxic molecules. Bryostatin is a molecule with a long history of toxicity. It was isolated from a bryozoan that I know has been repurposed. It was originally an anti-cancer lead, and I think now it's a neuropharmacological lead. So things you're looking for: one, typically lipophilicity, so you want something that can get through cell membranes often. Another key, and this is that's a reason I like the question so much, is that there are reactive parts to many natural organic molecules that lend themselves to protein binding. One is called a Michael system. So the best way I could explain that would be a double bond, a carbon-carbon double bond, which is conjugated to a carbon-oxygen carbonyl. So that's very reactive, especially to cysteine residues or serine residues that are common in many protein active sites. So there are researchers who specifically look for what's called a Michael system in molecules, because they know that that can be a good drug lead. One thing that we have found in the trichodesmium bloom material is a wealth of chlorinated metabolites, hundreds of chlorinated metabolites. Cyanobacteria have an underlying metabolism. So the way their genes are organized and the way their genes work, they so far have been the only organism that's been found to make, the trichophysin B had it, it's a double bond with that chlorine attached to it that sticks up from the main carbon chain. Technically, you'd call it a chlorovinylidene. And so that chlorovinylidene group's been found on many drug leads as well. And you can actually see that pattern. You can pick out if the molecule has chlorine in it and how many chlorines or how many bromines from the mass spec. So that's another really powerful tool in the mass spectrometer. So maybe, yeah, try to sum up. You typically want lipophilic molecules. You want them to be reactive because they're going to have to do something to some protein target. And many of them just happen to be halogenated in the marine realm. A lot of that's due to the amount of chlorine and bromine that you have there compared to the terrestrial realm.
Okay, good. I'm glad I wasn't sure how valid of a question that was at first, because I'm like, oh, can we just apply domoic acid into certain
I think with, I think one thing I didn't talk about with that cyanobacterial drug, that brentuximab that came from dolastatin. Now, dolastatin, if you were to take it as a drug, it would never work. It's far too toxic. You couldn't just take it intravenously. The way that works is they actually link it to an antibody. The antibody is very specific to a receptor on certain cancer cells. So the antibody is going to bind to that receptor. It's going to be taken into the cell. The toxin, that dolastatin, is linked to it. When it gets into the cell, that linker is cleaved by proteases that are in the cancer cell. It liberates that toxin, and now the toxin can kill the cancer cell. So there are a lot of biotechnology applications that are being used out there that are repurposing or are taking drugs that were thought to be too toxic, that were thought to not really subject well to the pharmaceutical world, and they're being able to solve those problems with medchem and with biotechnology.
I don't have another question, but I have a comment from David Borgman, who says that in reference to the temperature question related to the algal bloom, the greatest shellfish domoic acid concentration we have seen in Rhode Island was during March 2017, when the water temperature was between 2 and 0 degrees Celsius. So just a comment in that regard. But yeah, I don't see any other questions from folks. So if there's anything, oops, nope, I lied. I see one more. Are there similar cyanobacteria species commonly found in Narragansett Bay that you may look into for the next possible drug hits or leads? My question is, why travel to the Gulf? Do we have any cyanobacteria here to look into?
Yeah, another great, these questions are just fantastic. So I'll answer the last part first. So the reason that we actually went into the Gulf was that there was just one paper that had been published on trichodesmium, and they had collected those samples from the Gulf. And so that was the impetus to go there. And it also coincided with this ichthyoplankton cruise that NOAA goes on. So NOAA is going to sampling stations repeatedly every year, and they're looking actually at bluefin tuna larvae. That's why they go. And they're going to these, again, GPS located sites every year. So we piggybacked basically onto that cruise. We were really happy that they let us go on as volunteers. So that was kind of the reason there was the only other paper that had been really published on trichodesmium chemistry, where there were strains isolated from the Gulf, and because the ship was going out there. To the first question about Narragansett Bay, we've been wanting to do more in Narragansett Bay. We're just trying to find that time. One of the most fun thing, one thing I love about science and the whole community is that my grad student and I were at Riley. We were looking at a paper. It was from 1954 from the Journal of Botany, in which two scientists mapped out all the cyanobacteria in Narragansett Bay. And so we thought it would be really, really fun to go back to some of the sites that they had visited and to see what we could find there. But the specimens that they had found are well known for producing a lot of compounds. So we think that Narragansett Bay, one reason, I forgot I was going to try to include a line about how we do think that Narragansett Bay could be a great place to look for marine drugs.
Great. Again, believe that was the last question. If there's anything else that you or Bethany would like to add before we close this discussion. Oh, just kidding. Another question came in. The satellite map you showed of Lake Erie showed Toledo as a city with an accentuated bloom shadow. However, the right side of the map is clear. Is there any practice in Toledo that promotes this?
Yeah, another, keep them coming. These are fantastic. So I should say that I'm not an expert in the cyanobacterial blooms in Lake Erie, but from my reading and to the best of my knowledge, it happens to do with the concentration of pig farms in that part of the state and other farming in that part of the state. That's in the, I think it's called the Maumee watershed, maybe. There's a real concentration of farms, and the thought is that a lot of runoff from the farms leads to nutrient inputs that are leaving, I believe it is the Maumee River, I'm actually not 100% sure, but going out into Lake Erie. And so I think that's the point source, basically, why the concentration happens to be in that area. But I'm not the expert there.
I'm afraid to say we don't have any questions, because then one might pop up. But while we wait for any other questions to come in, are there any other final thoughts that either one of you would like to share?
All I can say is that I really appreciate the opportunity. Really nice to have an audience. I wish it could be in person, but I really enjoyed this. I hope this was my first time doing something like this, so I hope it was all right. And yeah, I'm just thankful for the opportunity.
All right.
There you are.
Mic unmuted. Thanks. First of all, I'd like to thank Rhode Island Sea Grant for the support and for the vision that this is potentially something worth keeping an eye on in Narragansett Bay following those events in 2016. And I'd also like to really highlight other people in the area doing the work. I know Dave's on this webinar, but Dave Borkman and Lucy Miranda also had a Sea Grant funded project and have been doing work out at some of the sites a little bit prior to us, doing more measurements and things like muscle tissue. And I think that our groups and the Department of Environmental Management and our team, everybody's sharing data and working together to try to solve this problem. So it's a really good example of collaborative regional science that I'm excited to be participating in. And I think it's been a great training platform for all of our students that have been involved as well. So we encourage everybody on this call to stay in touch with us. And if you have other questions to ask, we are always excited to reach out and answer them, and thank the audience for hanging in there. And we're so excited about these projects. Yeah,
that's just to emphasize that again. Yeah, Dave Borkman, Lucy Miranda, we met as groups beforehand. They gave us just great insight, and yeah, this is a really nice community of researchers. Not just environmental science, but in the chemistry world or in this area too. Yeah, absolutely.
Well, I want to thank Matt and also Bethany for all of your work that you've done and for sharing this research with us today, and for everyone attending. And we'll be virtual for a little bit longer, but hopefully we can continue these conversations even in these formats, because there's really good work happening, and I think it's very interesting and it's good for people to know. And like I said, the webinar is being recorded, and we will post that on our website, as well as send everyone an email notification once it's ready to go. So again, thank you everyone so much for joining us. And if you want to find out more information, just visit Rhode Island Sea Grant's website. And thank you to our supporters, and again to our researchers for all the work that you do.